Dear students, hello and namaskar. Myself SP Singh from Kendriya Vidyalaya Sangathan. This session is for class 12 biology students based on chapter human reproduction. This is second session in the series of 4 sessions and our focus will be on gametogenesis. The learning objective. After this session, the students will be able to draw a label diagram of spermatogenesis and oogenesis. You will be able to describe the formation of sperm and ovum and be able to differentiate between spermatogenesis and oogenesis. Let's proceed on with the session. First of all, we'll see the definition of gametogenesis. It is the process by which the primary sex organs, that is testes and ovary, they produce male and female gamete. To begin with, let us take spermatogenesis. This is the picture that we see in the book. Let's see how this picture is to be analyzed. Spermatogenesis in human male, it begins with puberty and proceeds in the seminiferous tubules that we did in our previous session. The peripheral layer of the seminiferous tubule consists of cells called as spermatogonia. These cells multiply by undergoing mitosis and increase in number. Some of these cells, they are ready to go in the next stage. They are called as primary spermatocytes. Now let us see how the primary spermatocytes behave. It is meiosis initiation. Meiosis 1, these primary spermatocytes which are actually diploid, they undergo meiosis 1 to form two haploid cells called as secondary spermatocytes. Now initiates meiosis 2. These secondary spermatocytes, they undergo meiosis 2 to form spermatids. And the next stage of this is spermiogenesis, where the spermatid undergoes change in their shape. They develop tail to form spermatozoa or the sperms. These sperms, then their head attaches to the Sertoli cells for nutrition that you can see here. Finally, the fully formed sperms, they are released from the seminiferous tubule and this process is called as spermiation. A total of 4 sperms are formed from spermatogonia. Now let us see how the hormones are affecting the formation of sperms. The first is gonadotropin releasing hormone. It is produced in the hypothalamus and it is increased level stimulates anterior pituitary gland to release 2 more hormones. They are follicle stimulating hormone and luteinizing hormone. Let us see the effect of follicle stimulating hormone. FSH, that is the follicle stimulating hormone, it stimulates Sertoli cells, as a result of which certain factors are released that will help in spermatogenesis. Let us see luteinizing hormone. LH acts on Leydig cells and they stimulate the synthesis and secretion of testosterone, which you know is very vital. It stimulates the process of spermatogenesis in males. Now let us see this particular picture. This is the picture of the sperm, spermatozoa. If we see in general, we have 4 sections that can be differentiated: 1, 2, 3 and 4. Let us see the details of the sperm. It consists of 4 sections, we have done it. The body is covered by plasma membrane. Now let us focus on the first part, that is the head. Head consists of anterior part. It contains male nucleus that has 23 chromosomes in it. Then it has acrosome at the top. Acrosome acts as a cap on the structure. Acrosome releases certain enzymes that dissolve the membrane of the egg and this helps in fertilization process. Let us see the next part, which is the neck. It is a small depression between first and the third part. It consists of centrioles, and these centrioles help in making tail and also helps in making spindle fibers. We move on to the third section, which is the middle piece. The middle piece, it contains numerous mitochondria that you can see here. These mitochondria provide energy for the movement of the tail. And the last part is tail. Tail makes sperm to be able to swim during the process of insemination in the female reproductive tract. Let's move further to the word that is written is semen. Let's see what is semen. It's milky white male genital fluid. It consists of sperms and the fluid secreted by all the accessory ducts and the accessory glands. That we did was epididymis, vas deferens, seminal vesicles, prostate and bulbourethral glands. Now let us see ejaculation. It is the release of semen by the male from this penis. One ejaculate contains between 200 to 300 million sperms. Out of these, at least 60% of them should have normal shape and size, and this is the normal shape and normal size. Out of that, at least 40% should be vigorous in motility, then only they will help in fertilization. The testicular hormones, that is androgens or testosterone, they maintain the function. They maintain the function of accessory ducts and the glands. Let's move on to the female gametogenesis. This is the photo in the book. It is familiar, you know it is ovary. Now within the ovary oogenesis occur. Let's study how the oogenesis starts. Oogenesis, it is the formation of female gamete. But things to remember is, in female, different steps are involved which are distributed over the years. The first step is from fetal stage till birth. Let us see what happens there. In the fetus, the process of gametogenesis, formation of gametes, the process of gametogenesis is initiated in the fetus itself. Nearly 2 million gamete mother cells are produced in each fetal ovary. These gamete mother cells are called as oogonia. Initiation of division. Each oogonia, which is actually diploid, it enters into prophase 1 of the meiosis. Then it stops further growth. This is called as primary oocyte. It is still diploid. The primary oocyte gets surrounded by certain cells, granulosa cells. Now it is called as primary follicle. Formation of primary follicle. Each primary oocyte gets surrounded by group of granulosa cells and it is called as primary follicle. From birth to puberty phase, a large number of primary follicles like this, they degenerate. Nearly 60 to 80,000 remain at the time of puberty. The next phase of oogenesis is from puberty onwards. Let us see what happens in this case. Formation of secondary follicle. Now each primary follicle that you see here gets surrounded by more layers of granulosa cells. This is called as secondary follicle. The next phase is tertiary follicle. These secondary follicles, they develop a cavity inside them called as antrum, and they are now called as tertiary follicles. The layers around the follicle, they increase in number, that is resulting into formation of external theca and internal theca layers. Now let us see how the cell inside it, the primary oocyte, it changes in the primary oocyte. In the tertiary follicle that we see here, the primary oocyte grows in size, it completes its first meiotic division, forming two unequal cells, a larger cell and a smaller cell. The next is formation of Graafian follicle. The tertiary follicle, it increases in size, it develops more fluid in the antrum, and now it is called as Graafian follicle. The secondary oocyte inside this develops a clear zone around itself called as zona pellucida. The next phase is ovulation. The mature Graafian follicle, it will release the ovum, and this occurs in the middle of the menstrual cycle. Always remember, the female gamete is still in its secondary oocyte stage. It has not yet completed meiosis II. Now let us compare spermatogenesis with oogenesis. If we see this particular table, spermatogenesis is a continuous non-stop process, but oogenesis, it stops at the primary oocyte stage before birth. Spermatogenesis is initiated only at puberty, whereas oogenesis, it is initiated at the fetal stage and then it stops. It is restarted at the time of puberty. Spermatogenesis continues late in the life, maybe beyond 60 years also, whereas oogenesis, it continues only till menopause. That's nearly 50 years of age. In spermatogenesis, the size of the cells formed remains same. The daughter cells, they are of the same size, whereas in oogenesis, the size is different. The daughter cells formed after meiosis II, they are of different sizes. One more difference that is very characteristic is, in spermatogenesis, polar bodies are not formed, whereas in oogenesis, they are definitely formed. What we see next is the tail. Sperms develop tail that makes them mobile, but in oogenesis, the ovum does not change at all. It does not develop a tail. And lastly, spermatogenesis, total 4 sperms are formed, whereas in oogenesis, only 1 ovum is formed and 3 polar bodies are formed. Always remember, whenever meiosis occurs, 4 daughter cells should be formed. In spermatogenesis, there remain 4, but in oogenesis, it is only 1 ovum and 3 are polar bodies. Let's see further. If we compare the picture here, you can very well make it out. There are certain sections which look similar on both sides, whereas there are certain sections which do not look similar on both the sides. Let us start at the stage 1. If it is oogonia in the oogenesis, it is spermatogonia there. Both are having 46 chromosomes, so they are diploid cells. They undergo division. Primary oocyte, you can see there, primary spermatocyte, same. The difference is only this thing, this phase is occurring at puberty, whereas this phase is occurring in the fetus. That is the only difference here. After birth, see the change when the meiosis occurs and the 46 is reduced to 23. At this stage also, the secondary oocyte, there is only 1 oocyte here and the second is the polar body, whereas on this side there are 2 spermatocytes. The difference starts here. This secondary oocyte, the 1 oocyte that we have, undergoes meiosis II to form again the final cell called as ovum, whereas in case of spermatocytes, each one makes spermatids, and then starts the main difference. The ovum, which is actually secondary oocyte, or we can call it as ootid, it does not change, whereas spermatid, it changes its shape From a globular shape, it becomes motile, having a tail, so that it can swim. So, in this particular picture, you can very well differentiate which sections are having major differences, which sections are nearly similar. But always remember, explain different stages of spermatogenesis and oogenesis. Compare and contrast spermatogenesis with oogenesis, and how many sperms or ovas will be produced if you start with 100 spermatogonia or oogonia. You can test it out. So, dear students, in this second session of reproductive system, we have focused on gametogenesis in both human male and female. I hope you all have enjoyed this session. I also hope that you have understood each and every aspect of the topic covered. In our next session, we shall meet again with the third episode of the human reproductive system and focus on menstrual cycle. Thank you.